Hello guys, welcome back to Joan Academy. In this particular video, we'll be looking at the various stages of erythropoiesis. Erythropoiesis has to do with the process of origin, development and maturation of erythrocytes, which we commonly call the red blood cells. Hemopoiesis or hematopoiesis is not the same as erythropoiesis. Hemopoiesis or hematopoiesis is the process of origin, development and maturation of all the blood cells. Remember that the blood comprises of various cells like the red blood cells, the white blood cells, and the blood platelet. But before we go into the various stages of erythropoiesis proper, let's first of all look at the various sites of erythropoiesis. When it comes to the site of erythropoiesis, I would like us to know that the site of erythropoiesis varies with age. In fetal life, erythropoiesis occurs in 3 stages. The first stage is the mesoblast stage. The second stage is the hepatic stage, while the third stage is the myeloid stage. The mesoblastic stage occurs in the first 2 months of intrauterine life. Here, the red blood cells are produced from the mesenchyme of the yolk sac. Hepatic stage occurs in the 3rd month of intrauterine life. The main organ that is responsible for the production of red blood cell in this stage is the liver, although the spleen and lymphoid organs are also involved. As for the myeloid stage, it occurs in the last 3 months of intrauterine life. In this stage, the red blood cells are produced from the red bone marrows and the liver also. In the newborn babies, growing children and adults, the red blood cells are produced only from the red bone marrows. Up to the age of 20, the red blood cells are produced from the red bone marrow of all bones, be it long bones or flat bones. After the age of 20 years, the red blood cells are then produced only from the membranous bones like the vertebra, the sternum, the ribs, the scapula, the iliac bones, and the skull bones. Also, the red blood cells are also produced from the ends of long bones. After 20 years of age, the shaft of the long bones loses the erythropoietic function. And the reason for this is simply because the shaft of the long bones becomes yellow bone marrow as a result of fat deposition. In a situation whereby the bone marrow is destroyed or fibrosed, the liver and the spleen may then produce the red blood cells. Collectively, bone marrow is almost equal to liver in size and weight. Also, the bone marrow is as active as the liver. Although the bone marrow is where all the blood cells, that is, the red blood cells, the white blood cells, and the platelets are all produced. But 75% of the bone marrow are involved in the production of leukocytes, which is commonly called the white blood cells, while only 25% of the bone marrow is involved in the production of erythrocytes, which is commonly called the red blood cells. Despite more bone marrows involved in the production of white blood cells, the white blood cells are less in number than the red blood cells in the ratio of 1:500. The reason for this is simply because of their lifespan. The lifespan of red blood cell is 120 days, whereas the lifespan of white blood cell is very short, ranging from 1 to 10 days. And because of the difference in lifespan of the red blood cell and white blood cells, the leukocytes are produced in larger number than the erythrocytes. This is solely for the required number to be maintained. There is no way we'll talk about erythropoiesis without the knowledge of stem cells. So what then are stem cells? Stem cells are primary cells that are capable of self-renewal and also differentiating into specialized cells. Hemopoietic stem cells are the primitive cells in the bone marrow, which produce the various blood cells we know. Hemopoietic stem cells in the bone marrow are known as the uncommitted pluripotent hemopoietic stem cells. The pluripotent hemopoietic stem cell is defined as the cell that can give rise to all types of blood cells. At the very beginning of the blood cells development, the pluripotent hemopoietic stem cells are not designed to give rise to a particular type of blood cell, and it is also impossible to know the particular blood cell that will be developed from these cells. Therefore, they are called the uncommitted pluripotent hemopoietic stem cells. There are only few numbers of these uncommitted pluripotent hemopoietic stem cells in adults, but the best source of these cells is the umbilical cord blood. The uncommitted pluripotent hemopoietic stem cells becomes committed pluripotent hemopoietic stem cells when they are designed to form a particular type of blood cell, be it the red blood cells or the white blood cells. The committed pluripotent hematopoietic stem cells is defined as a cell that is restricted to form one group of blood cells. These committed pluripotent hematopoietic stem cells are further classified into two types. We have the lymphoid stem cells and the colony-forming blastocytes. The lymphoid stem cells give rise to lymphocytes and natural killer cells, while the colony-forming blastocytes give rise to the myeloid cells. The myeloid cells are any other type of blood cells except the lymphocytes. When the myeloid cells are grown in cultures, they form colonies. That is why they are called the colony-forming blastocytes. Let's take a look at the different units of colony-forming cells. There are basically three types. We have the colony-forming unit erythrocytes, whose cells develop into erythrocytes. The second unit is the colony-forming unit granulocytes or monocytes. The cells of this unit give rise to granulocytes. Remember in our previous video on the white blood cells, we said that the granulocyte comprises of the neutrophils, the basophils, and the eosinophils. Also, this unit also gives rise to monocytes too. Finally, the third unit is the colony-forming unit megakaryocytes. The cells of this unit are responsible for the formation of blood platelets. There are some changes that is observed in the colony-forming unit erythrocyte as they transform to the matured erythrocyte. These changes include the reduction in size of the cell. The size of the cell changes from 25 micron in diameter to 7.2 micron in diameter. The next changes that is observed is that there will be disappearance of the nucleoli and the nucleus. Also, hemoglobin will appear on the cell. Finally, there will be change in the staining properties of the cytoplasm of the cell. Now let's dive into the stages of erythropoiesis proper. There are 6 stages of erythropoiesis altogether between the colony-forming unit erythrocytes to the matured red blood cells. The first stage is the proerythroblast stage. The second stage is the early normoblast. The third stage is the intermediate normoblast. The fourth stage is the late normoblast. The fifth stage is the reticulocyte stage. And finally, we have the matured erythrocyte as the last stage, which is the sixth stage. Let's take them one after the other. The proerythroblast is also known as megaloblast. It is the first cell gotten from the colony-forming unit erythrocyte. It is very large in size and has the diameter of about 20 micron. The nucleus of the proerythroblast is large and occupies the cell almost completely. The nucleus has 2 or more nucleoli. It also has a reticular network. Proerythroblast does not have hemoglobin yet. The cytoplasm of this cell is basophilic in nature. Proerythroblast multiplies many times and then becomes small in size in the next stage, which is the early normoblast. Synthesis of hemoglobin commences in this early normoblast stage, although the appearance of hemoglobin occurs only in the intermediate normoblast. The second stage, which is early normoblast, is a bit smaller in size than the proerythroblast. Its diameter is about 15 micron. There is absence of nucleoli in the nucleus of this stage. The chromatin network condenses in this stage too, and the condensed chromatin network becomes dense. The cytoplasm of the proerythroblast stage is basophilic in nature. Because of this, it is also regarded as the basophilic erythroblast. These cells then develops into the next stage, which is called the intermediate normoblast. In intermediate normoblast, the cell is smaller than the early normoblast, with a diameter of 10 to 12 micron. We can see that the size of these cells is steadily reducing in each stage, although the nucleus is still present in this particular stage, but the chromatin network shows further condensation. It is in this stage that the hemoglobin starts appearing. The cytoplasm of this stage is already basophilic, but because of the presence of hemoglobin, it stains both acidic and as well as basic stains. So these cells is called a polychromophilic or polychromatic erythroblast. This cell develops into the next stage called the late normoblast. The late normoblast is the fourth stage of erythropoiesis. Its diameter decreases further to about 8 micron to 10 micron. The nucleus becomes very small with a very much condensed chromatin network. This nucleus is known as the ink spot nucleus. In this stage, the quality of hemoglobin increases and the cytoplasm becomes almost acidophilic. Because of this, the cell is now called the autochromic erythroblast. In the final stage of late normoblast, just before it passes to the next stage, the nucleus disintegrates and disappears. This process is called the pycnosis. The other remnant is removed from the cell. The late normoblast stage develops into the fifth stage called the reticulocyte. In the reticulocyte stage, the cytoplasm contains the reticular network or the reticulum, which is formed from the remnant of the disintegrated organelles. The reticulocyte is also known as the immature red blood cell that is a little bit larger than the matured red blood cell. The reason why this cell is called the reticulocyte is due to the reticular network in the cytoplasm of the cell. Also, the reticulum in the reticulocyte stems with supravital stem. In newborn babies, the reticulocyte count is 2% to 6% of the red blood cells, which means 2 to 6 reticulocytes are present for every 100 red blood cells. The number of reticulocytes decrease during the first week after birth. Later on, the reticulocyte count remains constant at 1% of red blood cells, or below 1% of red blood cells. Whenever the production and also the release of red blood cells increases, the number of reticulocytes also increases. Because of the presence of the remnant of disintegrated Golgi apparatus, the mitochondria, and other organelles of the cytoplasm, it makes the reticulocyte to be basophilic. During this stage, the cells enter into the blood capillaries through the capillary membrane from the site of production by a process known as diapedesis. And finally, on the stages of erythropoiesis, the last stage is the matured erythrocyte. The matured erythrocyte or red blood cell is developed from the reticulocyte. The reticular network of the reticulocyte disappears and the cells becomes the matured red blood cells, which now have the biconcave shape, that is the right shape of matured red blood cell. Its diameter is now 7.2 micron. The matured red blood cell contains hemoglobin and lacks nucleus. It is important we note that it takes up to 7 days for red blood cell to develop and mature from proerythroblast, but it takes up to 5 days to get up to the stage of reticulocyte and 2 more days to get up to the stage of matured red blood cells. Alright guys, that is it for the stages of erythropoiesis. In our next video on blood physiology, we will talk about the factors that are important for erythropoiesis. Don't forget to subscribe, like and share this video. God bless you and stay blessed. See you next time.